Well, today we're going to talk about another class of membrane proteins. Last time we talked about proteins that are used to hold cells together, like the adherence junctions and the gap junctions. And this time we're going to be talking about another class of proteins, and these are proteins that are involved in the movement of molecules from outside of the cell to the inside of the cell, or vice versa, from the inside to the outside. Basically, transport proteins. And we can divide the transport proteins into what I want to say is two strategies. One is referred to as facilitated diffusion, and that is it's passive, and the molecules that are being transported transport along the concentration gradient. So the movement along the concentration gradient, there's two types. There's what are called ion channels, and there are transport proteins. Both of these move molecules or ions across membranes according to the concentration gradient. So if you look at the bottom of the figure, it shows on the right hand side shows the concentration gradient where the whatever molecules we're talking about are high on the outside of the cell and lower on the inside of the cell. And if you look over on the left-hand side, it shows basically diffusion, or what they call passive transport. So there's simple diffusion, like we talked about, some of the molecules that are polar, that are planar and non-polar, such as the steroid hormones, like estrogen or progesterone, they can just move through the membrane by simple diffusion, so they'd go from high to low. The transporters, or the ion channels, also move molecules according to the concentration gradient, and so you can see there's the ion channel being shown and then a transporter being shown. The ion channels, when they allow these molecules or ions to pass through, basically what's happening is they are allowing the free flow of transport. It's not actually transport, the free flow of these molecules or ions across the membrane, and it can be millions per second. When we talk about the transporter mediated or the transport proteins, usually it's according to the concentration gradient, but it will only move one or two at a time. So there's more of an active process, but still according to the concentration gradient. And then the second class is what we call active transport, which requires energy. And what happens with active transport, it's kind of also shown on that figure on the bottom, is that it moves compounds, ions, molecules against the concentration gradient. Like the pumps, when we talked about way long time ago, when we talked about electron transport, and the pumps pumped the molecules, in this case it was the protons, across the membrane, increasing the concentration gradient. So that requires energy to move these things and increase the concentration on one side of the membrane. Quite often the energy is ATP, but it can also be allosteric movements that are generated by other forms of energy. And in the case of electron transport, it was the movement of the electron through that molecule. In any case, I want to start by talking about the ion channels. So the ion channels are large transmembrane proteins, meaning they have multiple transmembrane domains. The sodium channel is a perfect example. It's a very large protein of over 1,800 amino acids, and it has 24 transmembrane domains. There are many different types of ion channels. They are extremely specific. So you will have a chloride ion channel, a sodium ion channel, calcium, potassium, and they're not interchangeable. So only sodium can pass through the sodium channel, only potassium can go through the potassium channel, and so on. So they're highly specific. They pass through what is called a hydrophilic core. Sometimes it's referred to as a water-filled core. And, and so what's the basis of that pore, or that hydrophilic core that allows the passage of these ions through the membrane? It turns out that hydrophilic core is generated by the transmembrane domains, but they are generated specifically by amphipathic alpha helices. So remember, we talked about amphipathic alpha helices, where you have the polar R groups on one side of the helix and the nonpolar R groups on the other side of the helix. So let's take a look at that. So here it shows the hydrophilic core. So this would be an ion channel. Basically, it can also be the gap junction, because they're made in similar ways, because it forms a core that allows the passage of polar molecules through the membrane. So what's showing here is, on the left-hand side, is kind of an example of the amphipathic alpha helix with the nonpolar on one side and then the polar groups on the other side. And so you can imagine that as these things come together and they form kind of a circle in the membrane, these transmembrane domains, the hydrophobic side of the helix is in contact with the hydrophobic interior of the membrane, and the hydrophilic side of the amphipathic helix is facing inwards, and so that generates basically a hydrophilic or a polar passageway that would allow water through and would also allow ions to pass through. So another use of the amphipathic alpha helix to generate these polar cores that pass through the nonpolar portion of the membrane. Now, they're not always open in order to allow the passage of ions through, and they can be open and they can be closed, and this is referred to as being gated. So all the ion channels are gated. They can be closed, and then under some stimulus they can be open. So this is illustrated in the next slide coming up. So here we have some illustrations demonstrating that ion channels are gated, and the gating refers to that they can be closed and then they can be opened to allow the passage through. And of course, these changes, the opening and the closing, the gating mechanism, are allosteric conformational changes in these proteins. Now, there's a number of different stimuli that cause the conformational change that allow these things to open and close. And so what they're showing up on top is the closed conformation. What they're showing on the bottom is the open conformation, where you see there's a hydrophilic passageway through the membrane. And so there's a number of different stimuli. There's what are called voltage-gated ion channels. These, of course, are used in nerve transduction, where you have a voltage differential that allows the passage of signals. Also, some of the cells in the heart are voltage gated. That's why you can restart the heart when you use those kind of paddles that they use that generate electric current and get the thing going again. Then there are ligand-gated channels, and these ligand-gated channels kind of make more intuitive sense to me. I can kind of visualize what happens a little bit better, because it makes sense to me how when you bind a molecule to, or a ligand to, a receptor, which is part of the channel, then that can cause an allosteric conformational change that would allow the opening of the channel. So the ligand-gated channel is another one. There's mechanically gated channels. That is the contact or some force on that channel will allow it to open up. So that would be like the sense of touch. When you touch something, the touch is actually moving and ion-gated channels, which allow the nerve impulses to begin. And also hearing. Hearing is generated by mechanically gated channels. That is, there's the little hairs in the inner ear, and they move due to the vibration, and then that movement opens these channels. It allows impulses to take place. There's also light-gated channels, and we see these in certain forms of bacteria, which as well as some other single-cell organisms like Chlamydomonas that actually also undergo photosynthesis, and the light is also involved in opening and closing particular ion-gated channels. So examples of open and closed channels, basically they are allosteric conformational changes, and this next slide just kind of shows that. So here's an example of X-ray crystallography that was performed on a closed channel. This is a ligand-gated channel, and on an open channel, and what you can see, so this is the X-ray crystallography, and you can clearly see that there are changes in the conformation of the protein, and these changes would allow the passage of these ions. So a lot of times you hear about, or you see drawings in textbooks where they'll talk about a gated channel having a kind of a flap that prevents the passage of ions, and then in response to either mechanical or ligand or voltage, then that flap opens up, kind of like a cap that opens and closes. And that may look a little bit crazy, but in fact that may actually be what's occurring in many ways. So you can imagine a alpha helix being turned in such a way as to create a cap or a flap that blocks the passage, or a beta sheet type structure which would be present, that would cause a closure or a cap on the one side of the channel, and then the allosteric conformational change would shift that in order to open it. So these are open and closed channels, and once they open, there is a absolute free flow of ions through the channel, millions per second. It's like the membrane is just open. Now, there's been a lot of attention on the ion-gated channels because it turns out that one disease, which has been very well studied, which is cystic fibrosis, is actually due to a defective chloride channel. And so channels kind of fell out of popularity for a while, and then when the gene for cystic fibrosis was cloned, and it became clear that it was some kind of a channel, and then later it was turned out to be an ion channel, a chloride ion channel, then it became much more in vogue to study these things, particularly the chloride channels, in order to develop drugs that would help alleviate some of the symptoms. So cystic fibrosis is due to a defective chloride channel. This is an inherited disease. It's actually pretty common, relatively speaking, in the Caucasian or European population. It's thought that 1 in 25 people are carriers of this mutation. It's recessive, and the estimates are about 1 in every 2,500 births, the individuals or babies have cystic fibrosis. So what happens in cystic fibrosis is that there's a mutation in the chloride transporter which prevents it from opening or prevents it from functioning. So chloride cannot pass out of the cell. So in a normal cell, which is shown on the very left-hand side, the ion channel, which is called the CFTR, cystic fibrosis transmembrane receptor protein, can be open and closed, allowing the transport of chloride out. What happens when chloride passes out of the cell, and this particularly affects lung tissue, when the chloride passes out of the cell, then sodium comes in to balance that charge, and then due to the high concentration of salt, the cells or the extracellular fluid is brought in to create a more fluid environment on the surface of the lung cells, and so the cilia, those cilia that are shown, can move due to the presence of the high amount of fluid due to the chloride and the potassium. What happens in cystic fibrosis, this is kind of shown in the middle, or on the one on the right, is that the chloride channel is non-functional. There's mutations that prevent it from opening, or in the case on the very right-hand side, there's a mutation that prevents the appearance of the chloride channel. And so what happens in these cases is, since the chloride cannot escape from the cell, and then the sodium does not come in to balance the charge of the sodium, so the fluid, the aqueous fluid that comes in to dilute out that sodium chloride in a normal tissue, then is no longer present, and so you end up building up this extremely thick mucus on the interior of the lung. And so what happens in these patients is that they get an enormous amount of kind of thick mucus on the interior in their lungs. They cannot clear the lungs of any bacteria, which would normally happen in a normal cell because it would just be very fluid. You can cough it out. This does not move, and so it gets very thick, and the cilia are no longer functional. And so these people end up, first of all, having a hard time breathing, and second of all, having a very difficult time clearing any kind of infectious material. So patients with cystic fibrosis get numerous repeated lung infections because that thick mucus buildup cannot allow the passage of the infectious agent out. So they get repeated lung infections. They can be cured by, usually by antibiotics, hopefully. But then the curing and the damage that is caused is they get a lot of scarring in the lung tissue. So over the course of years, they get more and more damage to the lungs. They have a much more time harder breathing, and eventually they can't really breathe anymore. The treatments for cystic fibrosis have improved. It used to be that somebody with cystic fibrosis would only live until maybe 10 or 12 years old. But now the treatments have improved and the therapies have improved. They have not cured it yet, although they're trying with gene therapy to replace and put the normal gene back in. But the treatments have improved, and so people can live well into their 30s and even older. So that's cystic fibrosis, a defect in the chloride channel. The other thing I want to mention is that if you look on the right-hand side, it says the common delta F508. That's a phenylalanine, a deletion of a codon, which causes the loss of phenylalanine at position 508. So here again we have an example of a single loss of amino acid which completely destroys the function of the protein because of the shape and everything we talked about earlier when we talked about proteins. But the interesting thing is that 70% of individuals with cystic fibrosis carry the same mutation. So why would that be? The hypothesis is that in some cases that there may be a selective advantage for carrying the defective gene. It's almost as if, and this probably is what happened, since so many people have that F508 mutation, that there's been a selective advantage that that particular mutation is spread through these populations. As if it arose once, it carried some selective advantage in the population, and it spread. It was allowed to spread by breeding. If you have something that's extremely lethal, that would generally eventually disappear from the population, but this spread to the population where we have 1 in 25 which are carriers. So the thought is, it's analogous in a way to what we see with sickle cell anemia. Sickle cell anemia in the black population is present as 1 in 13. 1 in 13 black individuals are carriers for the sickle cell anemia gene. What happens with sickle cell anemia is that there's a mutation in the hemoglobin gene, and so it causes the cells to change their shape. It's kind of a sickle-like form. They can't bind as much oxygen, and they also have a harder time passing through small capillaries. And so patients with sickle cell anemia have an enormously difficult time, enormous amount of pain. They have a hard time getting enough oxygen, and it's a very, very severe disease. But, and those are people who have 2 copies of the mutation cystic fibrosis, in sickle cell anemia. But as it turns out, if you're a carrier for the allele for a sickle cell anemia, that is, you have 1 mutation in 1 allele and the other allele is normal, you do not show the phenotype. You're a carrier, you do not show the phenotype, but it turns out that these individuals have a much more resistant phenotype to malaria. So it probably rose in Africa at some point. People who were heterozygous for the gene, for the mutation, were more resistant to malaria, and so there tended to be a selective advantage, and so it spread through the population to the point where 1 in 13 carry the allele. So in the heterozygous state, there's a clear selective advantage for maintaining this, even though in the homozygous state it's severe and could be lethal. So the thought is that the same is true, or something similar is true with cystic fibrosis, that the mutation, the F508 mutation, arose and spread through the population through some kind of selective advantage, that it gave some benefit by being a heterozygote. And the thinking is that it arose in Europe, and that individuals with the heterozygous state, that is 1 mutant cystic fibrosis allele, because the cystic fibrosis channel, this chloride channel, controls the amount of extracellular fluid, the thought is that people who are heterozygous, while they don't show the full cystic fibrosis phenotype, they still may have less extracellular fluid than people who are normal. And so the thinking is that it may have arisen and may have been a selective advantage against diseases like dysentery, for example, where you die due to dehydration through diarrhea and vomiting and so on. So they may have less extracellular fluid and would survive. In any case, that's the chloride channel cystic fibrosis. Now, When we talk about ion channels, we talk a lot about the flow of ions across, and generally to control charges on different sides of the cell. Also, in the case of the chloride channel, is to increase the negative charge on one side, so it brings in the sodium. But as it turns out, the ion concentrations and the flow of ions can have absolutely dramatic effects on cell physiology, aside from the charge differential. So, in addition to changing the charges on different sides of the membranes and changing the ion concentrations and the possibility of attracting more fluid, like we saw with the chloride channel, ion concentrations can have a dramatic effect on cell physiology, and the example is the most famous example is calcium. So the opening of the calcium channel, depending on the cell type, can lead to enormous changes in the cell behavior. It can lead to cell division, cell differentiation, muscle movement, neuron activity, changes in gene expression. So the flow of the ion generates all these various different phenotypes of cells. So how does it do that? It does it through calcium-dependent proteins and enzymes. That is, calcium flows into the cell and has an effect on the activity of all kinds of different proteins and enzymes, leading to cell division or differentiation, movement, and so on. But it happens specifically through a particular protein called calmodulin. So we're going to talk a little bit about calmodulin. Calmodulin is a calcium binding protein. So here's a drawing or an image of calmodulin. It's a crystal structure. Calmodulin has 4 binding sites for calcium. So when calcium flows into the cell, the calcium will be bound by a calmodulin. And then calmodulin turns out to be a regulatory protein for all kinds of other proteins and enzymes in the cell. So the calcium causes the binding to the calmodulin causes a allosteric conformational change. That conformational change then allows it to bind to other proteins and activate or inactivate them. So the example that's shown here is that the calmodulin, which is bound to calcium, is binding to a particular target protein, which is a kinase. And a protein kinase will phosphorylate all kinds of other proteins and modulate their activity. So here's kind of an overall view of this. So here we show at the top the cell membrane, the plasma membrane, with a calcium channel, calcium ion channel. And this particular channel would be a ligand-gated channel, so it's showing an extracellular signaling molecule binding to the calcium channel, opens up that channel, it's a gated channel, opens up the channel, and allows the free flow of calcium into the cell. Millions of calcium ions per second. Binds to the calmodulin. The calmodulin becomes activated through allosteric conformational changes, and then can bind to and affect the activity of hundreds of different proteins in the cell. So here it's giving an example of metabolic enzymes, so it alters the metabolism, cytoskeletal proteins, alters the shape or movement, transcriptional regulators, which then can alter gene expression. So you can imagine once you alter gene expression, then the entire behavior of the cell can change, like cell division. And this is only showing 3 classes: metabolic enzymes, cytoskeletal, transcription regulators. There could be transcriptional repressors that are bound, which would turn off gene expression. Could be translational regulators. So you can see how the entire physiology, the entire behavior of the cell can change by the flow of calcium. Now, interestingly, this can be done by one single extracellular signaling molecule, say a hormone or any other kind of a protein or growth factor that would bind to the calcium channel, so you can have one single channel opening up and have this complete change of activity of the cell. So this is referred to, and we'll talk about it later in more detail, this is referred to as a signal transduction pathway. That the signal, a single signal on the surface of the cell, is then fed into the cell, and the signal is transduced to the interior of the cell. One event at the surface activates many downstream proteins. It's also referred to as what's called amplification, that the signal is amplified as it goes on. So we start out with one signal, which is the binding of a signaling molecule to the calcium channel, some ligand opening the channel, and then the signal amplifies by binding to, lots of calcium comes in by binding to many calmodulins, and then those many calmodulins become activated and then bind to many effector proteins, and so the signal just amplifies and expands as it goes. Okay, so that's what I wanted to say about the ion channels. I want to talk a little bit about transport proteins. They also move molecules in the direction of the concentration gradient, so they don't require energy. It's more of a passive thing, but it happens one or two at a time. So in this illustration, it's kind of illustrating what these transport proteins are. They move molecules in the direction of the concentration gradient, but unlike the ion channels, which there's a free flow through the membrane once the gating is open, these things move one or maybe two at a time. So it's much more controlled, and you can think of it as the transport, the molecule that's being transported would cause an allosteric conformational change in the protein that would then open it up and allow the passage of this thing, but it's one at a time. So showing up at the top, glucose. So glucose has a high concentration on the outside of the cell, low on the inside of the cell. And so the glucose tends to enter. It binds to a particular binding site inside this transport protein. That causes a shift in the conformation, opens it up on the cytosolic side, and then allows the passage of the glucose into the cell. Now there's a number of different classes of these transport proteins. We'll start out on the right-hand side. There's what's called a uniport. That is, it transports one molecule at a time. That's like the glucose that we just talked about. There's called an antiport, which will transport molecules in two different directions. Again, driven by the concentration gradient. So the blue is going according to the concentration gradient, and the red is going according to the concentration gradient, just in opposite directions. So those are antiports, and then the symport drives two together, both in the same direction. Now some symports can drive a molecule against the concentration gradient. So everything we've talked about here, which is true, everything is moving according to the concentration gradient from high to low. But there are examples of symports which would move, let's say, the red according to the concentration gradient from high to low, and in doing so would move the blue from low to high. That is, the high would be on the inside of the cell. So the binding of the two together, the passage of the red allows the passage of the blue, even though the blue is against the concentration gradient. And this is shown in the next slide. Because what that implies is that there's energy associated with it. Whenever you move anything against the concentration gradient, it requires energy. And so the energy would be supplied by the high concentration of the red on one side, moving according to the concentration gradient. So let me kind of illustrate that in the next slide. So this is illustrated by what's called the sodium glucose symport. It's moving, the protein moves sodium and glucose in the same direction, that is, into the cell. But if you look on the left-hand side, the plus, that's the sodium, there's an electrochemical gradient where you have the high concentration of sodium on the outside, low on the inside, and so it would move according to the concentration gradient from outside the cell to the inside in the direction of the arrow. While the glucose, which is shown on the right-hand side, is a high concentration on the inside. So it would move, if it was moving according to the concentration gradient, it would move out of the cell. Well, what's happening here is that the sodium and glucose are both being imported into the cell. And so it's showing that in the illustration. So at the first figure on the left shows basically just the transporter inactive, and then the second one shows the sodium binding. And when the sodium binds, it allows the binding of the glucose. And when the glucose binds, it causes this conformational change that then empties both the sodium and the glucose into the cell. So what's really happening here is that there's active transport going on, because active transport moves it against the concentration gradient from low to high. And that's being done, basically the energy is being provided by the potential energy of the high amount of sodium on the outside of the cell. So while the sodium can move, in principle, the only way it really can enter is by binding glucose and allowing the transport. So the sodium gradient is potential energy driving the transport of glucose into the cell. Okay, so that's a form of active transport, but most of the time when we talk about active transport, we talk about energy that is generated by the hydrolysis of ATP, although this is, in a sense, active transport. Well, it is active transport, not in a sense. But the energy is coming from the gradient. So here's active transport, pure active transport, characterized by the sodium-potassium active transport pump. Like I said, always requires energy and often ATP. Although in the previous example, the energy was the potential energy of the concentration gradient of sodium. But in this one, both ions are being moved against the concentration gradient. So we have sodium is high on the outside, potassium is high on the inside, and every time there's a movement of the pump through the hydrolysis of ATP, which is the energy source, we get more potassium coming into the cell and more sodium going out of the cell, both of those moving against the concentration gradient. Now, all of these movements are allosteric conformational changes. So people have studied that and worked out what some of these changes, what some of these allosteric changes are and how they're driven by ATP or by the binding of sodium. In the previous one, we looked at the symport. The binding of the sodium took place passively, and the binding of the glucose induced the conformational change that then pushed it in. So this was going to end the discussion on these pumps and the transport across membranes, but let me just describe what's happening here. So this is the sodium-potassium pump, undergoes a series of allosteric conformational changes where it increases or decreases the affinity for either sodium or potassium. So we start out at the top left. What happens is in the empty conformation, there is a high affinity for sodium. So sodium binds to the pump. When sodium binds, it causes an allosteric conformational change, which causes the activation of a kinase, which hydrolyzes ATP and phosphorylates the pump. So it adds the phosphate. The addition of the phosphate induces another allosteric conformational change that opens the pump on the other side and lowers the affinity for sodium, and so it comes out. When the sodium comes out under lower affinity, there's actually a higher affinity for potassium in that state. So the potassium then binds. The binding of potassium causes another allosteric conformational change, which causes the dephosphorylation of the pump. In other words, the phosphate comes off. When the phosphate comes off, that again induces another conformational change that reduces the affinity for potassium, and so then it's released. So you can see what happens here. Binding of sodium, it gets pumped out. Binding of potassium gets pumped in against the concentration gradient. But all these are controlled by these allosteric conformational changes. And in some sense, the addition of the phosphate uses the energy from ATP is really only one induction of a conformational change. The other inductions of conformational changes come from the release of sodium or the binding of potassium or the release of the phosphate. So all these are allosteric conformational changes. Okay, so that is pretty much what I want to say about the transport. So we talked about the ion channels, which can be gated. They can open and close. Once they're open, there's a free flow of ions across the cell membrane. That could lead to changes in charge distribution, and it can also lead to important changes in cell physiology, particularly with calcium, which then allows the activation of all kinds of other proteins through the calmodulin. Then we looked at the transport proteins, which move things really one or two at a time, so it's much more controlled. And then we looked at active transport, which requires energy. Generally speaking, it's ATP, which causes conformational changes. But remember, active transport can also be generated by the energy in the gradient in the case of sodium. So it's a potential energy, still energy. Okay, have the good rest of your day. And the next time we will be talking more about signal transduction.